Hey guys, in this video we're going to learn about binary fission. So that's the special process bacteria use to make copies of themselves. So here are our slides from chapter 8 on Blackboard. And in this video we're going to cover these objectives. We're going to describe the process of binary fission. And then we're going to name some organisms which divide in this way. So we've already said that the prokaryotic cells, like bacteria, use this process to divide. And remember, bacteria have just one chromosome, and remember, they don't have a nucleus. The prokaryotes do not have a nucleus, but they do have one chromosome that's in a loop in the middle of the cell. And remember, we said this chromosome is sitting in the nucleoid region, which is a bit confusing for people because they start confusing this with the nucleus. But remember, a nucleus is a special compartment that your DNA sits inside. There's no nuclear envelope. There's no wrapper around this genetic material. It's just free inside the cell in this nucleoid region. So the genetic material of your prokaryote is going to be in its nucleoid region. Now, the prokaryotes are going to use this process, binary fission, to divide. So remember, binary means two, and fission means splitting. If you're trying to do atomic fission, you're trying to split the atom, right? So fission means splitting up, fusion means trying to stick things together. So we're doing binary fission, splitting our cell into two copies. So when you're doing binary fission, remember, all the copies you make are going to be identical to the original cell. So sometimes they say all the daughter cells are original or identical to the parent cell. But this starts confusing people because then they start thinking that bacteria has male or female sexes, which it doesn't, right? A daughter cell is just this term that's used to refer to any offspring from that parent cell, that first cell that you have. So it has nothing to do with sex, male or female. So here's my original prokaryote with the chromosome. And remember, they have one chromosome that's in a loop inside the cell. So the first thing we're going to do is make a copy of that chromosome. So we're going to replicate the DNA. So now here's our original chromosome, and here's a copy. So we made a copy of the DNA. So DNA replication means make a copy of the DNA. And then we do cytokinesis, which is a special process that means that you split into two cells. So you're splitting the cytoplasm of this big cell into two separate cells. So you can see the plasma membrane and the cell wall. Bacteria will have a cell wall as well, pinching inward to separate this into two individual cells. And then we'll end up with two identical daughter cells at the end of the process. So we started with one cell and we ended up with two. And the key thing here is they're identical because we made an exact copy of the DNA during this process of binary fission. So here you can see a picture showing you how it works. So this is my prokaryote with its loop of DNA inside. It's one chromosome in a loop inside the nucleoid region. So the first thing you do is you make a copy. So you enlarge the cell and you make a copy of the DNA, the genetic material that's inside your cell. So here we started off with this chromosome that was colored red, and now we've got this additional blue colored copy inside our cell. So notice that it's got this special area where the chromosome sticks onto the plasma membrane and the cell wall. So each chromosome attaches to the cell wall in a different area of the cell. So it's kind of getting pulled to two opposite poles. So as the cell stretches out and grows larger and starts to move apart, the chromosomes are pulled in opposite directions as well because they're anchored onto the cell wall. And then finally you do your cytokinesis, where you pinch in your plasma membrane and your cell wall to try and separate the two cytoplasms off the cell. And then finally they'll separate, and now you'll have two independent cells. But the key thing here is that these are genetically identical, right? So sometimes when bacteria copy, we refer to them as clones. Clones means that they're all the same, right? So clones are going to be genetically identical. So because you're just directly copying the DNA and because you're making an exact copy of that chromosome, all the cells that are produced by this process are also going to be genetically identical. So that's the process of binary fission. And like we said, that's the process used in prokaryotes to make copies of themselves.